morning. I want to thank the organization and the organizers for promoting my abstract to speak today, and for the organization for their support over so many years of so many scientists. Mirit Adaljem has beaten me yesterday to it to being the least recent awardee to speak at this meeting. So it's 30 years ago that I got a long-term postdoc fellowship. And because of my advanced age as a mid-career scientist, I permit myself some general remarks here and there. And one is that one of the great features of being a scientist is that you have mentors, and also you're always giving advice to the younger generation. So I had a pretty good set of mentors and continue to do so. On to science. There was a pointer here. This? Oh, okay. No, it doesn't look like a pointer. Ah, okay. My lab is interested in the biophysics of protein-protein interactions as the molecular basis for mechanisms in cell signaling. Specifically here, ligand binding to a receptor outside the cell. How is this information transmitted to the inside, and then what happens here? So two proteins interact. What are the mechanisms, and how does this lead to specific functions? We're interested here in features of the protein, such as the interacting surface. You open this up and then look at the character of this surface here for this little protein pair. One side is entirely positively charged, the other one is negatively charged. We're interested in conformational changes that occur when proteins interact, but also changes in their dynamics. And then these interactions take place in particular environment here near the cell's plasma membrane. So why do only these two proteins interact and not the rest of the cell as well? The methods we use is primarily solution NMR, crystallography, computational modeling, and a lot of molecular dynamic simulations, as well as biochemical assays. The biological problem is in cell migration. So this is an example of axon guidance. Developing neuron here sends out one axon. This is guided here at the end by a growth cone, which really needs to make a decision whether to move towards the target cell, form synapses, and so forth, or whether to retreat from this environment, collapse back, and move off in a different direction. How is this done? The guidance cues, these are molecules attached to the target cell membrane or secreted by them. And in the growth cone, then there's several families of receptor proteins. We study the plexin family here. Neuropilin is the co-receptor in the F receptor family. The beautiful thing about these receptors is that they're used all over the place for many different processes in development, the immune system, cardiovascular, bone skeletal, the list goes on. Here's an example from zebrafish embryo trunk, where the ligand sets up fields, the semaphorin here, and the plexin D1 in this case makes sure that developing blood vessels can just move between these segments. When you interfere with the plexin D1 signaling, you get this uncontrolled sprouting. So while normally the signaling is repulsive, sort of keep out, don't go here, you can switch it around to a more attractive or invasive response. And this happens in some cancers. Actually, for plexin, all these are co-receptors, famous receptors involved in cancers like MET, EGFR, or B2, and so forth. How is the growth cone moving around? This is the small GTPases of the Rho family that then modulate the actin cytoskeleton and create forward and backward collapse. I don't have time to talk about small GTPases, classical molecular switches. We had quite a run of papers to show how their signaling is affected by even transient interactions with the plasma membrane, papers here on RAS GTPase. How does this work with plexin receptors? So it was noticed early on that there's, on the intracellular side here, there's homology to GTPase activating proteins split into two segments by the insertion of a polypeptide chain that binds Rho GTPases specifically. And we started this project back in 2002 when I began my lab at Case Western by determining the solution structure of this segment here. This is a superposition of the main chain fold, and then was a cartoon. Turned out to be related to the structure of ubiquitin, just with longer inserted loops. And that actually made it very difficult to identify that it was ubiquitin-like from alignment. You just couldn't get the sort of loops right until you knew the structure. We then asked how does this domain interact with the Rho GTPases, and we localized the interaction to this region here, and this is a RAC1, one of the Rho GTPases. The typical switch 1 and 2 regions are interacting over here. But how does the dynamics, the internal fluctuations in the protein change when these two interact? And we saw here that there was some rigidification, but also more flexibility on the side of the plexin domain. But on the side of the RAC1 GTPase, we had quite long-range rigidification of the structure. You just saw less mobility, and that's significant because really on the opposite side of the binding interface here, the protein becomes less dynamic, and then we think is now able to bind other interaction partners, because the entropy penalty that needs to be paid on binding is already kind of prepaid. In the RBD, we identified some pathways for the protein structure dynamically and allosterically, but I don't have time to go on to that, except I want to show a little part of molecular dynamics movie here, which suggests that those two structures, rather than being stuck together firmly like you would infer from a crystal, are really wobbling around quite extensively. And so the most persistent contacts are only formed 40% of the time in this movie. And that suggests a very dynamic picture of those interactions. We can do a sort of network analysis, see how the two proteins talk to one another, and there's not much interaction here across the interface in the case of RAC1. But with the homologous Rho GTPase round one, you see quite extensive protein-protein crosstalk. We had another system in the lab between a SAM domain forming a heterodimer, and this had even more extensive changes, really was able to stick together in quite different configurations. And Rose came up with the idea that they're really sort of a principal interface that is used by many binding partners and also alternate orientational binding partners. And this speaks to molecular economy and cell signaling, perhaps. The alternative would be that there'd be new alternate interfaces, say over here and over there, but then unoccupied interfaces are typically bad news in kind of protein aggregation and amyloid formation and so forth. Why would we want to have different configurations of these complexes? While near the membrane, some of these would be disallowed, for example, due to geometric restraints. So protein complexes dynamics makes sense. I want to tell one short story about the other part of the plexin intracellular region here, the GTPase activating protein, which has the RAP1 RAS protein as a substrate. And we noticed that when we do an activity assay here, the GTPase activating protein helps to the GTPase to hydrolyze the GTP to GDP and phosphate here. We detect the phosphate by fluorescent binding protein. As a function of substrate, the RAP1B concentration, the plexin just suddenly becomes much more active. This is a nice hill S-shaped curve. Cooperativity happens. So how does this arise? And through simulations and experiments, we found that there's a loop here in the plexin structure that can either beat down and occludes the substrate binding side, or is folded back and interacts with the juxtamembrane region here, which also undergoes a conformational change. Now, when the loop is down, there is slow turnover. It basically has to flip up before RAP protein can bind. But in the case there is a lot of substrate around, every time a RAP protein has been sort of processed and dissociates, another one can bind again. And so this becomes much more efficient than this low concentration RAP1 situation over here. And this is referred to as allostery. It's a kinetic cooperativity effect. Finally, the major unsolved questions about these systems still are around the plasma membrane. So again, how does the signal binding, ligand binding outside get communicated to inside the cell? And these are type 1 receptors, so they just have one polypeptide segment, a helix typically going across the membrane. And the understanding is that most of the time receptors dimerize and bring the extracellular domains, of course, together through the ligand binding. The transmembrane regions form helix dimers, and then intracellular domains also come together. But within such a helix dimer in the membrane, you can also have configurational changes. You can slide the helices, you can change the crossing angle, rotate them, and this becomes interesting because several states must exist even within a dimer, often active and inactive states. One can design transmembrane-like peptides that will either interfere with these active dimers or in fact stabilize them. And we have a collaborator in Tennessee, Francesco Barrera, who's been looking at this, and in Strasbourg, Dominic Bagnard. We turned to coarse-grained simulations a number of years ago, purely because of the amount of computing time one could save. So rather than representing each atom with a point in the whole atom molecular dynamics simulations, we've grouped atoms together here in the side chain of amino acids and represent them by particles, give them particular properties, and that's a big saving in computing time. But also the energy landscape is somewhat more smooth, so there's another acceleration factor of 10 to 50-fold. So overall, these simulations run orders of magnitude faster than the all-atom ones. So we tried to predict what these helix dimer structures would look like across the membrane. We also have the lipids here, coarse-grained, and the waters. We start off the system as two ideal helices with some amino acids at the extracellular and intracellular part is an extended configuration. Then they drift together and form these helix dimer structures after a number of microseconds. We did this for 11 systems where the structures had been determined by NMR spectroscopy, mostly in bicels, but some in detergent, and then compared the coarse-grained results to those NMR structures. And the results are not that great. So there are some which do very well. Here, 1.4 angstrom, RMSD 2.2, 2.8, but many are above 3.5, 4 angstrom even. So the coarse-grained force field is not really good enough at discriminating the packing of helices that is required. And there's often a sliding of helices. Now, our surprise was when we used Alphafold, here Alphafold multimer, and here we got some very good results, 1 angstrom or less even. So 9 out of the 11 structures had less than 3.5 angstrom prediction. And many of you will have read about Alphafold and come across it. These are some slides from the literature. There's some systems that works very well for folded protein domains, not so good, more extended structures with long loops, and then intrinsically denatured states are pretty ugly. And all this is a slide we're putting together for perspective. Depends on what structures and information you put in here into kind of the tree trunk, the software, machine learning, and AI to go into the branches. Here, monomer predictions are pretty accurate. Multimers are coming along. Protein design is making some progress, but other things are sort of in the clouds, as it were. And I'll skip a few things. The major issue I have with this is that it's a black box, like many machine learning AI things. So does Alphafold understand physics? I think the jury is still out. I talked to some experts, and I think it hasn't really shown the emergent properties that AlphaGo and others have shown already. So is not understanding what it's doing, how it really works, a problem. And David Baker in the webinar asked him that question, and he thought, well, we've been living fine with our own neural networks for millions of years, so should be fine. But I would say that, no, as scientists, we want to understand and abstract rules and then tinker with them. And but classical molecular dynamics is a major force still. So Anton 3, I think, is about to come to the Pittsburgh Supercomputing Center, commuter, sorry. And it's really hundreds of times faster than the biggest other supercomputer summit, for example. And we're looking forward to using it. Final slide, lessons after 30 years or so. Time and productivity are not linear. Luck and considerable effort are involved. Academic life is hopefully long. Change is overall good. I recently did a Master's in Business Association degree, learned a bit about other things. And one key piece of advice I asked when I started my lab, Art Palmer, where I was visiting at the time at Columbia University, said, whatever you do, don't assume that others should think like you or act like you. Everyone is on their own journey, and I want to thank outstanding lab managers and postdocs. About half of them got senior academic positions over the years. It's a global village of collaborators, and I'll leave you with a summary. Thank you for your interest and attention.
Thank you very much for that overview of your 30-year career. Thank you. Questions?
All very clear. We can chat at coffee then.
May I just make a quick comment? Guys, you may have noticed that we have alumni talks, and this is something we'd really like to continue going forward. that we also bring back our alumni into the active Awardees community. And that's why, which makes my job difficult when putting up the program. So that's why we will continue with the poster talks so that we can also integrate our alumni into our meetings. That's why. Thank you for that.
Thank you.